Welcome to Biology 210, Lab 12 Genetics, Part 2. We will begin with exercise 36, the effects of UV light on bacterial growth. This is on page 317 of your lab manual. So we're going to begin a discussion with a little background on ultraviolet light. Ultraviolet light is also known as UV light, and we primarily think of UV light coming from the sun or tanning beds. UV light can not only alter bacterial DNA, but it can also alter our DNA and cause cancer. So let's talk about what UV light is. Light in general travels in waves or wavelengths. The wavelengths of light are measured in what is called nanometers, NM, nanometers. We've discussed that in the metric system back at the beginning of this course. Shorter wavelengths of light have more energy. So for example, X-rays and gamma rays. They have more energy because as the light travels and they're shorter, they lose or dissipate less energy into the atmosphere. This also makes these wavelengths of light more dangerous to work with because they have more energy. Longer wavelengths of light have less energy. Again, that is because as the wavelengths travel, they lose more energy because they have to travel a longer distance. An example is UV light. It is not uncommon to have laboratory hoods have a light source as UV light that actually acts as a disinfectant. In other words, when you turn on the light in the hood, it actually cleans the work surface. There are three kinds of UV rays: UVA rays, UVB rays, and UVC. UVA range from 315 to 400 nanometers. UVB rays range from 280 to 315 nanometers. These are the two that you look for on your sunscreens or sunblocks. They can cause skin cancer and sunburns. UVC rays are the rays we will focus on this class. UVC rays range from 100 to 280 nanometers. And you should note, and it is significant, that in this experiment, UV light that are used are set at 254 nanometers, which is the nanometer that causes the most damage to bacteria, specifically bacterial growth, and we will see why on the next few slides. Page 318, 319. Let's talk about the setup of the experiment. So before we begin, there is a very good pre-lab that actually walks you through the setup of this experiment. It is slightly different than ours, only in the times. We use slightly different times, but the rest of it is exactly the same. So step 1, a total of 8 nutrient agar plates are swabbed with bacteria. 4 of those nutrient agar plates are swabbed with bacillus. The other 4 are swabbed with Staphylococcus epidermidis. So before we look at the next few things, let's think about this. First of all, what is the variable in this experiment? Good, the types of bacteria. We also will use time as a variable as we continue through the steps. So why did we choose Bacillus and Staphylococcus epidermidis? Staphylococcus epidermidis, as you should recall, is a microbe found or bacteria found on your skin. Bacillus is a bacteria we've worked with quite a bit this semester and is a very unique property that you should recall from previous exercises. Step 2. Plate 1 is exposed to UV light at 250 nanometers for 1 minute, plate 2 at 3 minutes, plate 3 for 5 minutes, plate 4 for 7 minutes. So we actually have a total of 8 plates, but we're going to set up 4 of them, 1 for each bacteria. So in other words, Bacillus, plate 1 is 1 minute exposure, and Staphylococcus, plate 1, is 1 minute of exposure. Each plate has half of the plate covered with an index card. The purpose of the index card is to act as a control. So that side of the plate should have lots and lots of growth because there's nothing to keep the bacteria from growing. We're giving it the food, the temperature, everything that it wants. We are only concerned with the experimental or exposed side of the ultraviolet light. We then add the ultraviolet light for the amount of times given. We incubate the plates at 38 degrees for 48 hours and view the results. Again, the pre-lab video has a very good demonstration of the setup They just use things like 30 seconds and 1 minute, and we use an extended period of time. But the example is exactly the same. So how does UV light decrease microbial growth? In other words, how does this actually damage the bacteria? You should recall that we have different kinds of bases, A's, T's, C's, and G's. The base that we are going to talk about the most is called thymine. It says causes thymine dimers to form, and you're saying, what is a thymine dimer? Di means two, so basically what happens is it causes two thymines sitting next to each other to form a bond. That bond is not supposed to form, and when that bond forms, it causes problems. If that bond forms, those thymine bonds, then that acts as kind of like a little speed bump. And because of the speed bump, RNA polymerase cannot bind. If RNA polymerase cannot bind, then transcription cannot occur. If transcription does not occur, then translation cannot occur. No translation means no enzymes or proteins are produced, and therefore no metabolism. Without an enzyme, you cannot perform things like glycolysis, the prep step, the citric acid cycle, etc. That means no ATP is produced, and the cells are going to die. The cells cannot divide. So on figure 36.2, we can see those dimers forming. The more dimers that form, the less growth, meaning the more this is really mutations, the more mutations that occur. So the arrows show us a nice little picture. So exposure to UV light causes thymine dimers, no replicating DNA strand, which means no transcription, translation, or metabolism. Page 320. On page 320, fill in the purpose of this experiment. On page 320, shade or draw on the amount of growth hypothesis for each plate, the control and the exposed side. Here's a hint. Your control side should be the same for all 4 plates. You only need to draw it for one bacteria. So I think there's just a set of 4 plates. So draw it for either Bacillus or Staphylococcus. What you should draw on the exposed side is as the amount of time increases, so 1 minute, 3 minutes, etc., what do you think is going to happen to the amount of growth? And then also think about comparing and contrasting Bacillus to Staphylococcus. But again, you only need to draw in for one type of bacteria. This page will be submitted on Blackboard. So here we have the data. We are now on page 321. Here's the results for Bacillus. So it says on page 321, fill in the table only. So you don't need to draw the pictures, just the table with negative dash, meaning no growth at all, plus, meaning a little growth, 2 pluses, a lot of growth, or 3 pluses, the most growth. Again, you're only looking at the exposed side, and you only need to fill in the table for one set of Bacillus. So there's 3 Bacillus and 3 Staph. Only need to fill in 1. So let's look at the control side. So on the control side, the squiggly lines that I have created represent lots and lots of growth. On the exposed side, we have 1 minute with quite a bit of growth, 3 minutes with less growth, 5 minutes with less growth, and 7 minutes with even the least amount of growth. So the plate with the most growth would have the most pluses, and then you would go down from there. So that is the results for Bacillus. So do you think the results for Staphylococcus epidermidis are going to be the same? Think about the properties of Bacillus. Staphylococcus epidermidis results. So again, you're going to fill in the table for one set of data for Staphylococcus. You're only worried about the exposed side. So again, 1 minute has lots of growth, 3 minutes less, 5 minutes no growth, 7 minutes no growth. That would be dashes to represent no growth. So was there a difference in the amount of growth for Staphylococcus versus Bacillus, and why would that be? Page 322, conclusions and analysis. On page 322, answer the questions. You will be submitting page 322 on Blackboard. Exercise 37, genotype versus phenotype using Serratia. Page 323. So what is a genotype? The sum of all of the genes. Your genes is your exact sequence of nucleotide bases. Again, what is the order? ACC or GGG or ATCG, whatever that order is. The phenotype is the observable set of characteristics that can change depending on the environment. So for humans, we get our genotype from our parents, half from our mother, half from our father. Our phenotype is what is expressed, turned on. So for example, if your mother has blue eyes and your father has brown eyes, that is your genotype, what you get from your parents. The phenotype is what we see in you, so maybe you have blue eyes. We also need to talk about a pigment called proteogyptogen, and what that pigment is a red color. It produces a red color under certain conditions or certain environmental conditions when this gene is turned on or expressed. So at certain temperatures, this gene is turned on and we see this pigment color of red. Page 323, the experiment setup. 2 nutrient agar slants are aseptically inoculated with the bacteria Serratia marcescens. 1 nutrient agar slant is incubated at 25 degrees Celsius. The other agar slant is incubated at 38 degrees Celsius. After 48 hours, the results are viewed. So what is the variable in this experiment? Good, it is temperature. On page 324, complete the purpose and hypothesis. Submit page 324 on Blackboard. So do this before you see the results on the next slide. Page 325, the data. So here are the results after 48 hours. As you can see, the Serratia marcescens on the left at 25 degrees Celsius produces that red color. Therefore, the gene for proteogyptogen production was turned on. The Serratia on the right at 38 degrees has a white color. So those genes were repressed or remained turned off. They were not expressed. Page 325, conclusion and analysis. On page 325, answer the questions. You will submit page 325 on Blackboard. Exercise 38, DNA structure, page 327. Read page 327 and understand the structure of DNA. We are not doing pages 328 through 329, the DNA extraction. That is something that's difficult to do if you're not physically in the lab. There are no pages to submit on Blackboard for this exercise. Just read the few paragraphs on the structure of DNA. Exercise 39, genetic transfer, page 331. Read page, it should say 331. That's a typo, so it should say 331. And understand the different types of genetic transfer. We are not doing page 332. There are no pages to submit on Blackboard for this exercise. You only need to read page 331, which talks about the different types of genetic transfer, things like transformation, conjugation, transduction. Be able to compare and contrast the 3 types of genetic transfer on page 331. So what are the assignments for this lab? Submit the required lab manual pages on Blackboard. If you haven't already, view the vaccine calling the shots video. The link is located on Blackboard. Those pages, which is the answers to the questions that correspond with the video, are also located on Blackboard and will be submitted. And then take quiz 8, which is all of the genetic information that we've covered in these 2 video tutorials. Contact your lab instructor if you have further questions.